Hello, everyone, and welcome to today's webinar, A-TEEM Tools for mAbs, Media, and Protein Identification and Separation Challenges in Biopharmaceutical Analysis. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by HORIBA. To learn more about our sponsor, please visit horiba.com. I would now like to introduce to you our speaker for today's webinar, Dr. Karen Gall, Software Product Manager, HORIBA Instruments Incorporated. For a complete biography on Dr. Gall, please visit the Presenter tab at the left of your screen. Now, today's presentation is interactive, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. Dr. Gall, welcome. You may now begin your presentation.
Thank you. So I'm Karen Gall. I'm Dr. Gall from HORIBA Instruments in Piscataway, New Jersey. And today I'm going to talk about A-TEEM tools for mAbs, monoclonal antibodies, cell media, and protein identification and separation challenges for biopharmaceutical analysis. Thank you for joining us. First, an overview of what we'll talk about today. I'll give an overview of A-TEEM spectroscopy and the benefits of this method. Then I'll give a few examples of applications for biopharma, some history of the A-TEEM method and how it was first used, and some new exciting work that we have to present on more protein-based therapeutics. Instrumentation and data acquisition tools for A-TEEM. So I'll talk about what we have at HORIBA and the data acquisition software that we have as well. And last, I'll present a new multivariate analysis software that has a guided workflow for analyzing A-TEEM data specifically. I also want to point out first what to look for from this webinar. So what you can take away from this webinar today. And so first, I hope you can all better understand some of the basics of A-TEEM. If you're already familiar, a refresher never hurts. How A-TEEM can fit into your current process of if GMP is required. So think about the ways that A-TEEM could possibly help you if you're working in a GMP environment or in a pharmaceutical environment. And understand a few applications of how A-TEEM is currently being used as well, to think about how if they apply to what you're doing in your own lab. And then to learn about the tools needed for adding A-TEEM methods to a GMP lab or process environment. Okay, what is A-TEEM spectroscopy? A-TEEM stands for absorbance, transmittance, excitation, emission matrix. To understand A-TEEM, let's first talk about what a fluorescence emission spectrum is. A fluorescence emission spectrum is where we excite a sample using a single wavelength and measure the photons emitted at higher wavelengths. At longer wavelengths, lower energy. An excitation emission matrix, is when we collect an emission spectrum at different excitation wavelengths across the absorbance range of a sample or a set of samples. And this gives you a series of emission spectra like you see here. And looking at these spectra in a different way, we can then plot the excitation wavelength versus the emission wavelength versus the intensity, which is an EEM, and it's often represented as a contour plot or heat map like this. So we now understand an EAM, but what is A-TEEM? So for A-TEEM, we collect the excitation emission matrix, but simultaneously, as we're stepping across the excitation range, we also detect the light that's transmitted through the sample. And from this, we can get information on the absorbance spectrum or the transmittance spectrum. And so we can use that data in two different ways. First, the absorbance spectrum is actually an orthogonal data set. There's a ton of information in the absorbance, just like, for example, color. You can look at the absorbance of all the absorbing species as opposed to the only fluorescence species from that sample. And then the second one is to use the absorbance to correct the fluorescence spectra, the fluorescence EAM, for inner filter effects. And I'll talk about this a little bit in a future slide. But this correction from the absorbance data enables the EAM to be measured more accurately over a wider range of concentrations than it would otherwise. A-TEEM contour plots can then be used to identify components in complex mixtures spectrally. So this is really an alternative to chromatographic techniques. It actually rivals chromatography in sensitivity, but it's also less expensive, and it has advantages in speed as well. And so it really is an optical spectroscopy method that can be very useful in regions where other techniques in chromatography or even other optical techniques, have gaps. So to talk about the inner filter effect a little bit, so here at the top is the equation with the corrected fluorescence and how we use the observed fluorescence and the absorbance values at both the excitation and emission wavelengths for each data point in the contour plot or the matrix. And so, so many samples can be measured with a regular excitation emission matrix. The A-TEEM, which is corrected, is more accurate for samples at higher optical density. And really, many, if not most, products in biopharmaceuticals are really working with higher optical densities, unless you're looking at some low contamination species. So here is an example we use just to show an example for a standard. This is quinine sulfate at both a low concentration, an optical density of 0.08, so below 0.1, and one that with a higher optical density of 0.49. So when we don't use an inner filter effect correction, this is fine. The shape of the contour plot for the E-TEEM, with or without the IFE correction, is accurate. It's the same. But at a higher optical density, for the uncorrected E-TEEM, you see in the lower left side, this one has a spectral shape that's skewed. Okay, and so this is skewed mostly in the excitation direction, in the excitation wavelength direction. But when we apply the inner filter effect correction using that collected absorbance data, we now have an accurate representation of that fluorophore's fluorescence fingerprint, even at higher concentrations. Okay, so HORIBA has a lot of content on inner filter effect corrections and how we use it with the A-TEEM method. And so if you want, you can look at all of those application notes and technical notes, and those are on our website if you want to learn more. So the inner filter effect really happens in two different ways. We have primary inner filter effect and we have secondary. And primary IFE happens because the light from the excitation source is shown in blue here. Here's the square. We're looking down at the top of our cuvette of a solution. And so the center is the focal point of the excitation light. The blue is the excitation light coming in. And the emission beam path is collected at 90 degrees orthogonal to, 90 degrees from the excitation. And so the primary inner filter effect happens if the solution here is too optically dense. That light doesn't reach the center of the cuvette, and so the fluorescence that's detected is actually detected as less than what it really would be, as linear to the concentration of the fluorophore. The secondary IFE effect is when the molecules that are emitted from an optically dense solution are actually reabsorbed by the solution because it's too strong, too optically dense. And so again, you have an emission profile that is less than what it really would be relative to the concentration. In the lower right here, you see a plot of the total fluorescence intensity measured versus the absorbance. And so without the inner filter effect correction, the inner filter effect actually makes the fluorescence signal non-linear with the absorbance or the concentration, and it should be linear. And so with the inner filter effect correction, now we have an accurate representation of fluorescence intensity up to higher optical densities. Here we can measure with a 1 centimeter path length cuvette optical densities of up to 1, absorbance of 1. But you can actually go higher if you use shorter path length cuvettes and geometries. And so this is a really kind of cool thing that we've done, is to be able to more accurately and quantitatively measure samples using a fluorescence technique with this type of correction. I want to look at A-TEEM now as a comparison of with different molecular spectroscopy techniques, with different optical techniques. And the space that A-TEEM fits now is really based on the benefits of sensitivity and speed, right? Optical spectroscopy is just faster than maybe chromatography. In comparison to other optical techniques, there's differences where A-TEEM can be used where others really kind of fail. And they're not necessarily competitive techniques, but we can use A-TEEM, we can use fluorescence to fill the gaps where things like vibrational spectroscopy, Raman, FTIR, and near-infrared, and even UV-Vis absorbance spectroscopy alone can fail. So for complex solutions and low-level ingredients, we have a much lower detection limit using fluorescence because it is such a sensitive technique. With A-TEEM, we can go even lower in accurate concentration than we can with EAMS. And those can be used for relatively complex solutions as well. A-TEEM is a fluorescence method and really will ultimately shine when you're looking at smaller molecules and lower concentration solutions. But with that said, A-TEEM does have usefulness in solid applications such as like cosmetics and skin analysis. And so you can also think about what other tools can and cannot do, and whether a sensitive optical technique like A-TEEM could fill some of those gaps. But overall, A-TEEM, the message is that A-TEEM has sensitivity and it also has speed. And so this can come in very handy for a lot of applications. All right, so let's look at some ways that A-TEEM can be used. The A-TEEM method really started with the water research community, and leaders in water research studied something called chromophoric dissolved organic matter, or CDOM. And this included molecules, fluorescent molecules like amino acids, fulvic acids, and humic acids, and other things that are found in all sources of water, from environmental water sources to water that's been treated with different processes. And many biological molecules do fluoresce in nature. And so these include amino acids like tryptophan and tyrosine and phenylalanine, NADH or NADPH, chlorophylls, different variations of algae. And so CDOM researchers and even water treatment plant monitors use EAMS and A-TEEMs, those fluorescence fingerprints, to separate and identify levels of these molecules in water. Okay, so this is really where the history of A-TEEM kind of started and grew up to be used. And the method is still used for water applications, but it turns out that A-TEEM is really good at looking to fingerprint other complex mixtures like wine, so from water to wine. And so any really food and beverage products, natural products, and more. And so for this example, you can see A-TEEM is used to measure final wine products. You can measure raw materials like grapes and grape extracts, or the final product. And this one used parallel factor analysis, also called PARAFAC, which is a multivariate analysis method to distinguish the 5 different components in a series of wine samples. And this was actually wine samples taken in wines that were manufactured in different countries. And so you can then see that principal component analysis, you can then classify the wines from the A-TEEM data to very, very well group where that wine came from. Okay, and so that comes from the different fluorescent chemicals that are in those wines. But you can actually take the extracts from grapes as well to predict which wine will taste and what the quality of that wine could be. So again, we have applications on this too. But what about pharmaceuticals? So water and food are great, but how does this matter for pharmaceutical labs and bioprocess and GMP labs, which are mostly development, manufacturing, et cetera. So the A-TEEM spectroscopic technique really comes down to the separation of complex mixtures. And so this can be applied to many things. So whether it's food or it's pharma or biopharma, such as small molecule APIs, raw materials, formulations, delivery vehicles, so things like AAVs and exosomes, cell culture and bioprocessing, those are very complex mixtures, protein therapeutics, contamination, and even drug construct binding. And so you can think of these things as mixtures as something like your favorite sugary cereal. One of my colleagues, Dr. Lu-Fei Chen, always refers to solutions in bioreactors as sometimes as soup. And so we have, you can also think of it as a soup. And so this may be a little bit more appropriate for some of you that are attending today. In any case, any of these areas really have a need for separation of complex mixtures, identification or quantification of individual components within them. And so you can think of A-TEEM as an optical technique for separation science. And so it can be used for QC checks, can be used to monitor a process, whether it's for concentration or classification reason. So I'm going to talk about a few of the biological applications that we've been working with at HORIBA using the A-TEEM method. So first, A-TEEM is really well suited for bioprocess monitoring, where cells are grown in media for production of some biological target product. And so here, this data is shown from a project with Innovate UK, Center for Process Innovation. And A-TEEM is used to monitor the bioprocess steps from cell media to the bioreactor phase, to flow, different flow-throughs, and wash steps as well. And because the fluorescence A-TEEM is based in fluorescence spectroscopy, as I said, it's both sensitive and it's selective for each step of the process. So you can see how your solutions are changing as you go through the different steps of the bioprocess. From that data, so these are actually Parafac loadings, and you can see that we're tracking, and this is the emission spectrum. No, this is the excitation spectrum on the left. In the middle is the emission spectra loadings, and on the right is the object loadings. So as the in-process samples go, you can see it went from cell media to bioreactor to the protein A flow through, wash steps, and a post measurement. And so each of these components, tyrosine, tryptophan in different local environments, and even vitamins and cofactors, you can look at the spectral profiles of those that are extracted from the parallel factor analysis, and then see how those change over different steps of the process. And you can actually see in step 5, which is the final wash step, that none of those biological molecules are there, which they shouldn't be. That actually makes sense. Okay. And so for bioprocess monitoring, the variation in cell media is also very important. It has noticeable impact on cell growth rates. And so A-TEEM contour plots, such as the ones shown here, can also be sensitive to those variations, which is very useful. And so by using A-TEEM, a cell media, the growth feeders, whether it's, you're putting in more glucose or vitamins, those decisions can be made on whether they are needed to be replenished or supplemented based on the indicators of the cell growth. And you can track those through A-TEEM. And principal component analysis, again, can also be used for the cell media characterization. The cell media quality and consistency is also of really critical interest. So when you're starting a bioreaction, you want to know that your cell media is of the right quality in order to get the product that you need. And so A-TEEM can be used to let you know what the variation is for the different cell media batches or from commercial suppliers, so that checks can be done to ensure that cell media is of good quality before it starts. Okay, and I have Yeah, okay. So this is the score plots for the PCA to show different batches from the same manufacturer. You can see how they vary slightly as well. So you can make decisions on what to start with your process based on your cell media. And here you can also see that cell media can change over time and have temperature effects on quality. And so this PCA shows a cell media batch over different days, but also stored at either cold or ambient temperatures. And so you can see that the A-TEEM data plot at day 0 and at day 5 at room temperature, by eye, they don't look all that different. But when you use multivariate analysis to separate or classify these data sets, you can see that the day 0, which is the light blue here, is grouped kind of far away from the day 5 at room temperature, which is the very top. I think I don't know if that's blue or black square dots, which are somewhat different in their quality. And so the PCA can then distinguish the media that's changed based on time and storage temperature. And so again, this can help you make decisions on which cell media to use to get good results in the bioreactor. Okay, and then this one has this slide is also showing results from the work we did. Again, this was through the Catapult project in the UK for cell and gene therapy. And this work was carried out as part of that product, the PAT consortium for that project. And you can see that the A-TEEM was actually tracked with different, with a bioreaction, and can be correlated with very well with the total viable cell number, which was measured with an external method. And so the A-TEEM, you can see in the two that are showing tyrosine, tryptophan, and NADPH, which can be related to cell proliferation. Okay, so those trend comparisons, they correlate very well with the cell number and the growth rates as well. And I have a short video to show just the process over some changes. Okay, so it's just a short video, but to show when the cells are growing, the A-TEEM is also changing. The next application is also an interesting one. And so we can use A-TEEM spectroscopy to tell very similar protein-based products apart, different APIs. And really, this is a tool that can be applied for QC checks. You can do a quality control check to make sure that your drug is matching what you think it is, or even the detection of drug counterfeiting in the forensic sense. And so this study was done using 7 commercially available monoclonal antibody drug products, the APIs. So each of these 7 have very similar protein structures, but different applications. And so as you can see by the A-TEEM fingerprints, and we put a dotted line guide because they all look very, very similar. Some of them differ you can tell by eye, they're slightly different in width. Maybe the location is a little bit dipped below that line or a little above. But by eye, it's kind of difficult to distinguish one from the other. But when we use multivariate analysis, we can take that data and actually separate them very easily. So the reason for doing this study is to look to see if a counterfeit drug is reverse engineered. The monoclonal antibody protein, which is the API, if that counterfeit one is very similar to the original, how can you quickly verify a drug's authenticity, right? Is it the real one or is it fake? So first, looking at these two, there's Imfinzi and Yervoy. And so the A-TEEMs by eye, again, they look very, very similar. In reality, the protein structures are also very similar, but they differ by one tryptophan residue and one tyrosine residue in their sequence each. Okay, so even though the fluorescence signature looks very similar, when you do a simple normalization and subtraction, you can actually get a difference A-TEEM plot that tells you that these are not the same thing. When you apply multivariate analysis to a bigger data set, you can then classify them very easily as well. And so you can see from the score plot of just the PC1 versus PC2 that 3 different drugs of very similar sequencing, but that have slightly different number of tryptophans or tyrosines show up in different regions of the score plot. Okay, so multivariate analysis using PCA can confirm that difference as well. For these 4, these actually have similar protein structures, the exact same number of tryptophan and tyrosine residues. So those are the fluorescent molecules. But those tryptophan and tyrosines, the fluorescence signature, the fluorescence spectrum and A-TEEM spectrum is sensitive to the local environment. So even if the differences are in a different part of the protein, they're not necessarily a difference in the the tryptophan and the tyrosine numbers. If it's a slightly different sequence in a different region, the tyrosine and tryptophan fluorescence is going to be sensitive to that, and it will shift slightly. And so even these 4 have varying between 70 and 90% sequence similarities. Those differences can be detected easily by A-TEEM. And as you can see in the PCA score plots, the PCA 1, 2, and 3, that you can classify them really very easily. Okay, so this can tell you if your drug is real or not. And so here's just another data. So this is for all 7 of them. So when you take all 7 of those very similar monoclonal antibodies that are different drugs used for different things, you know, there's really a lot of the variance can be explained by doing PCA. When you look at the ParaFAC, when you have the discrimination analysis where you can look at the components in the EAMs, you can see the pure tryptophan. There's 2 tryptophan-like components and 1 tyrosine-like components. And so this is looking at tryptophan in slightly different local environments based on the protein sequence. And the same for tyrosine. So again, you can separate these very easily. And the last application I'll talk about is protein aggregation. So protein aggregation, whether it's wanted in your solution or unintentional, is something that A-TEEM can also detect. And there's a paper that was published some years ago, 2013, and there's been a few more recently in the past few years, showing that some proteins, when aggregated into amyloids, so structured aggregates, those structured aggregates can actually photoluminesce kind of like semiconductors. And so they actually have a band gap type behavior, giving rise to fluorescence in the visible region. So tryptophan and tyrosine are typically absorbing and emitting in the UV region, right? They absorb around 280 nanometers, and tyrosine emits around 305, and tryptophan's 340, 350 nanometers. When you have the amyloid fluorescence, you actually start to see fluorescence in the 400 to 500 nanometer range, depending on the protein. And so you can see visually, the solutions don't look different. They're still optically pretty clear. But when you look at the A-TEEM data, you see that structure can grow. So this is an example of this Yervoy drug product. And you can see the protein aggregate fluorescence coming up. And over time, it's actually increasing based on the number of aggregates in the solution. So whether aggregates are wanted or probably not wanted, you can look at the quality of that solution to tell how much aggregate is present using the A-TEEM fingerprint. Okay, so this one also has a short video. This is over, I think, 100 hours with the increasing amount of amyloid formation. So that you can, let's see, here's the video. So you can see the fluorescence from the amyloids here, and this is just growing over time. And so using that fingerprint, you can then do quantitative analysis of the number, the amount of protein aggregates in your solution. Oh no, what did I do? I just closed something. There we go. Okay, sorry. Right, there we go. Okay, so the next one is instrumentation and data acquisition tools for A-TEEM. So HORIBA has what we call the A-TEEM compliance package, and this includes the instrumentation, the IQ-OQ validation procedure. So this is a documentation procedure. And we have new EasyPat OPC-UA server. So this is acquisition software if you need 21 CFR Part 11 compliance for the FDA and GMP. And we also have A-TEEM Director, which I'll talk about, which is the new multivariate analysis software. First is the A-TEEM spectrometer. This is called the Aqualog UV800. We also have an auto sampler shown here, and we have a 10 milliliter version for vials of 10 milliliters or 2 milliliter vials for smaller volume samples. And this is used if cuvette-based measurements just are not practical for your lab, if you have a lot of solutions you need to go through. The way that the Aqualog works is slightly different than a standard fluorimeter. And so we have both fluorescence and absorbance detectors. This image is looking down into the sample compartment, and this is showing a standard 1 centimeter cuvette holder. But we also have temperature-controlled sample holders, which are preferable for pharmaceutical and protein applications. For this diagram, you can see that the excitation light is coming from a xenon arc lamp. So the xenon lamp is the excitation light source, and the wavelength that is hitting the sample exciting the sample is selected by a grading position. So you scan the excitation grading across the excitation and absorbance range. And as we do that, we're detecting the light that's transmitted through the solution with the absorbance detector. So this measures the absorbance of the sample as we're scanning the excitation. But also, as we're scanning the excitation through this and through the sample, the fluorescence light that's emitted isotropically, so this is in all directions, is detected at 90 degrees to the back of the instrument, of the sample compartment direction. And we use a scientific CCD to collect the emission spectrum all in one integration. So on each excitation absorbance wavelength, we're also collecting an emission spectrum very quickly. Yeah, so this can be done down to about 5 milliseconds for one integration. So most A-TEEM measurements take less than a minute to collect, depending on the integration time and the step increment. It could be as quick as a few seconds under the right conditions. Okay, and so again, we use that absorbance spectrum to then correct the fluorescence so that you can get a properly corrected, IFE-corrected A-TEEM. The next part of the compliance package is the IQ-OQ document. This document's a complete installation checklist and operational checklist for installing and testing the operation of the instrument. It's done by a certified service engineer on site with the end user, and both the service engineer and the end user, which is you, approve this on successful completion of each step. Okay, so this is already available from HORIBA, but it's required by anyone that uses the instrument in a GMP environment. So I wanted to point this out. It is available. We have it, and it can be used for A-TEEM and for the Aqualog. The IQ-OQ procedures include the validation of the Aqualog spectral data outputs as well. And so since the Aqualog uses 2 different spectroscopy methods together, we validate both. And the IQ-OQ procedure follows, it's actually the OQ procedure, follows United States pharmacopoeia procedures on spectrophotometer performance for UV visible absorbance, and also the methods for fluorescence spectrometer performance. And we use NIST certified standard reference materials as required by these test procedures. All those passing results are then reported using the current software tools dedicated to test these procedures. And this has always been available on the Aqualog, but I wanted to point out that it's a necessary process for ensuring the instrument is operating as expected. So that covers the instrument and the validation procedures. So what about the acquisition software? So how can we collect A-TEEM data with everything that's required for GMP and FDA 21 CFR Part 11? And this includes the audit trail, data authenticity and data integrity, and also attributable records. And this includes electronic signatures. And as a bonus, it should fit into current pharmaceutical process analytical technologies, or PAT software. And a lot of pharmaceutical companies already use these big PAT enterprise softwares. And so for this, I wanted to talk about OPC-UA. The next part of the compliance package is really how HORIBA can offer a solution that enables GMP and enables 21 CFR Part 11. EasyPat is the OPC-UA server for the Aqualog, and it's been developed for the Aqualog using the software architecture that was first developed for the software called EasySpec, and this is the software that we use to control the Duetta spectrophotometer fluorimeter from HORIBA. And EasyPat is a little bit different than our normal software products. It's really just a server, and it's based on OPC-UA communication protocols. OPC-UA stands for Open Platform Communication Unified Architecture. It's a set of standardized protocols for machine-to-machine communication. And the important part of this is that OPC-UA has the security built in that enables that data authenticity and data security, so that 21 CFR Part 11 can be enabled. Really, a lot of the PAT software products that are commercially available act as a client to connect with different analyzers, different softwares, different libraries, LIMS databases, and analysis packages that have OPC-UA servers on them. And so the operator at a site can interact with the client software only, and then be able to control the process from data acquisition to analysis to electronic signatures, and also data security. The good thing about OPC-UA communication is that it can be used for the Aqualog. It can be used now to basically plug into a PAT software client, which is already in use. And so, as you may know, the processes used in PAT involve many methods other than A-TEEM. And so orchestration software often controls all of them. And so this light blue box at the top is meant to map out a possible PAT software orchestration software as the client. And we can communicate things like acquisition commands, errors, method prompts, error reporting, and most importantly, the data that comes to and from the Aqualog back into the orchestration software. Okay, so the EasyPat OPC-UA server really allows that interoperability, I love that word, with PAT software platforms that are already in use as enterprise solutions. Yeah, and so all of that with the PAT software, the operator really runs the orchestration. A manager or supervisor can define the method and control all of the orchestration setup, where the operator just runs it from a single interface. And so the server allows the Aqualog to just be plugged into PAT in that way. The EasyPat OPC-UA server is available to connect to, or will be available soon to connect to the different PAT software is already in the industry. And so Syntiq from Optimal Technologies and SciPat from Siemens are two that we can already, or that we are in the process of working with, so that those two platforms can build adapters to connect to the Aqualog. LabVIEW from National Instruments also has an OPCUA toolkit. This is a little bit more of an academic, but you can build a client interface to connect with the Aqualog as well, to call the acquisition functions and then pass the data into an orchestration or multivariate analysis platform too. So just remember, if you use one of these, you have to have the client that's available to connect with the OPCUA server called EasyPat from HORIBA. If you know of another company that uses another PAT software and can connect with OPCUA, you can bring those opportunities to the fluorescence and the life science groups at HORIBA. And we are very willing to go through that process, to work with that other company to see if we can be able to build a client and a connection with that software. And so we can be involved in supporting the development of the connection if you need it. It really is the beauty of using OPCUA, because it's a common and very documented communication protocol, and instruments from all different vendors can do the work together through this common process with these platforms. And so this also makes it a lot easier to apply GMP for these types of methods. And lastly, I want to talk about the multivariate analysis software that can be used for A-TEEM data. So we have the instrumentation, we have the validation, and we have the acquisition software. And so for the next thing is analysis software. And so what is needed? For most, not all, but for most of the A-TEEM applications, we do use multivariate analysis or chemometrics. So what is needed in such a tool? We need validated algorithms. We need data traceability at each step, because we need GMP and user login security. And it should be easier to use, right? Good software is easy to use. So the final part of the compliance package is called A-TEEM Director. A-TEEM Director is a new multivariate analysis software. It's based on Vector Director, which is a product that is sold by the KAX Group. This is KAX Group in Australia. A-TEEM Director will be licensed by HORIBA, but the platform was designed with icon-driven graphical menus. You can see the big toolbar on the right side with the icons. So there's a lot of graphically driven menus and easy to follow data analysis processes. For the pharmaceutical industry, A-TEEM Director specifically provides login authentication and a full audit trail throughout the analysis process. My favorite thing about this is that it has a user-guided workflow. So this is very specific to the process of analyzing A-TEEM data, and it takes you through working with three-dimensional data sets, which is very different or can be very different than working with two-dimensional data, to be able to get the data in, to do visualization of it and pre-processing of it. And A-TEEM Director really makes it easy for novice users to, one, keep going in the right direction through multivariate analysis modeling, and two, get the answers to the questions that you're asking from your data. And with all of the functions for GMP, this enables Part 11 compliance as well. So in this one, the user-guided workflow that's in the left corner here provides those step-by-step. Oops, let me go back. But provides the step-by-step guide on how to work with the A-TEEM data. And information windows are also available at each step. So if you have questions on exactly what that window is meant to do, or what the options are, you have information windows that you can turn on and off to keep going in the right direction. The guided workflow really brings you through what can often be a back and forth process between working with three-dimensional data sets, unfolding them, and working with two-dimensional data sets, and then going back and forth. And so it's a benefit to bring in the data and move through the process in a clearer way. And so I'll go through the guided workflow step by step with an example to show how you can help A-TEEM data analysis just become a little bit easier. But the guided workflow, as you can see, goes from importing the data, defining class variables, plotting and defining ranges, pre-processing the three-way data, and applying the parafact analysis, which is specific to three-way data, and then converting the three-way data to two-way data and going through the same types of processes with models like PCA or partial least squares regression or discrimination. And we also have prediction tools. For the file import, getting the data, especially three-way data like EAM or A-TEEM data, into a multivariate analysis package can often be one of the biggest steps, right? Just getting your data in. And so A-TEEM has a file import for HORIBA for the Aqualog specific data files. These are the .dat ASCII format files for the processed EAM data or EAM A-TEEM data, and also for the absorbance spectra that are exported automatically from the Aqualog software. The graphically driven menus and easy heat map visualization are helping you see the data, so you can see the fingerprints for all of your samples. It also includes all of the metadata read in from the Aqualog log files. So the way that each of those data sets was collected is brought into the software so that you can then trace your data points or your data sets back to how they were collected. The addition of classification and concentration variables, what we often refer to as Y reference data or Y block data, is as easy as copying and pasting them into a table. And so this example was from a data set that my colleague, Dr. Jeff Julian, collected of mixtures of the 3 different fluorescent amino acids, so tryptophan, tyrosine, and phenylalanine. And we can look at the classification of those mixtures and also the concentrations for each of the amino acids. Once we do that, we can then move on to data visualization and the range selection. The profile tool enables you to click on your EAM or A-TEEM contour plot and see the profiles in both the excitation and the emission ranges. You can also look at the average or mean of all of your samples, have a visualization of that, and then graphically select the region that you want to use for modeling. So this allows you to exclude any unwanted peaks or regions of the contour plots or background data that doesn't have any useful information in it. And you can use those ranges for all the modeling steps going forward. We then have pre-processing for the three-way data. So taking that data, you can apply, for three-way data, you can apply Rayleigh masking, which removes the scatter peaks, and also Raman masking. So if you have any solvent Raman peaks that are showing up in your data, you can mask those as well. You can change the area widths of those masks and then save those as a template, again, to be used in your modeling later. So if you want to try different types of pre-processing, you can make different templates and then apply each one to a model to see how the results can change. For parallel factor analysis, this one uses three-way data specifically, right? And so the guided workflow shows the step-by-step options for this type of model, including data selection, range selection, the pre-processing templates that we created, and any constraints on the model. Parafact is usually looking at only positive values, not negative ones, and algorithm parameters such as the number of components. And so for this example, we are working with 3 components, 1 for each amino acid in the mixture. And in the results of the Parafact model, the loadings for modes 1, which are the samples, 2, which is the emission spectra, and 3, which is the excitation spectra, are plotted. And we have error reporting such as residuals, and those can also be viewed and exported from plot options menus. But loadings can also be displayed in either spectra for modes 2 and 3 that we saw before, or in a heat map. And so here, if you look at the loadings for factors 1, 2, and 3, you can see the pure heme components for tryptophan, tyrosine, and phenylalanine that are used in the model, or sorry, resulting from the model. And the workflows here are similar for principal components analysis and partial least squares, whether regression or discrimination, which use the supervised Y block data that you input earlier. And these can be used to assign either classification groups or concentration for your data. There are also workflows to take models created and then apply them to incoming new data for classification or regression prediction. And so this once again uses the step-by-step process to select the new data for prediction, choose your Y variable data to associate it with, and then select components and the model to apply it with. And the guided workflow covers the options there from start to finish. So you start with importing your data, you create your models, and then you can predict new data using the models you created. And aside from the easy to follow guided workflow for A-TEEM, A-TEEM Director enables compliance. And so this includes user login and authentication that is managed by the Windows Active Directory. You have audit trail for all steps of the analysis, and the original data is always maintained. You can clone the data and work on that and make changes to that, but each step is tracked in the audit trail, and the original data always stays intact. So with all of these, this enables GMP and Part 11. I should also mention that the model algorithms used in A-TEEM Director are referenced, validated, and developed under GAMP software development lifecycle, which is important for pharmaceutical and process scientists who want to be sure of the analytical tools that they use. And so we ensure that those are, again, referenced and validated and tracked. And so with the A-TEEM compliance package, the instrument, the qualification, installation, and operational qualification, with OPC-UA for the EasyPat server, which fits into existing PAT softwares, and A-TEEM Director multivariate analysis, we can now really say that the door is open for using A-TEEM as a tool in GMP labs. And so in summary, today we've talked about the definition of A-TEEM, some useful applications in the area of biopharmaceuticals from history, and to some new exciting work that we've been doing at HORIBA, working on protein-based therapeutics and solutions. We talked about the instrumentation and the data acquisition tools available for A-TEEM, and about multivariate analysis tools for A-TEEM as well. And so I really hope that you can all take away from this thinking about what A-TEEM spectroscopy can possibly do for you in your lab and your applications as well. And with that, I want to thank Dr. Lu-Fei Chen and Dr. Jeff Julian for their work on the applications that were presented in this presentation. And I want to thank you and LabRoots, and I'm happy to take any questions you may have.
Dr. Gall, thank you so much for your informative presentation. It is now time for our live Q&A portion of the webinar. Now, to our audience, if you haven't already done so, please submit your questions. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. So, Dr. Gall, we have so many questions already coming in. I'm just going to dive in.
Okay.
So let's start. Let's start here. Can you detect bacterial contamination in bioreactors with A-TEEM?
Bacterial contamination. So I'm trying to think if we do it. So there are, I believe we do have customers that are looking at bacterial contamination. I don't know if they're specific for bioreactors. So that is something that we should look at in terms of an A-TEEM method development. I don't see why not. Bacteria do have UV fluorescence associated with them. And so I think that it's very possible. I don't know specific examples that we have, But if you get in touch with our applications team, I'm sure we'd be happy to talk about it and any experience that we have with it.
Thank you. Next question. If I need an aqualog that accomplishes CFR 21, do I need the A-TEEM director? Is that mandatory?
Right. So if you are working in a lab that requires 21 CFR Part 11 compliance, any of the data acquisition, you will need software that enables compliance, but for the analysis as well. So if you have results, you're doing analysis on the A-TEEM data, it's important to have the traceability, the user login, the data security and electronic signatures. So whatever Part 11 compliance or GMP practices you have in place in your lab, A-TEEM Director is a solution that enables that compliance. Okay, so there are other multivariate analysis tools available in the market. HORIBA works with many of those, but for the compliance, there's not always they're not always compliant. And so you do have to evaluate what tool is the best for you to use in terms of your own compliance process. Yeah.
Thank you. Now, Dr. Gall, can you comment on A-TEEM's applicability to highly complex substances mixtures or different media types, where you may generate a library of single substance spectra and use that to identify specific substances of interest or attempt to characterize the exposure of components that are solubility limited?
Okay, so let's see if I understand this question. You can't highly complex substances mixtures. Yes, A-TEEM can do that. Different types of media. So we get asked about A-TEEM libraries quite a bit. For other types of spectroscopy, Raman, FTIR, NIR, it's all about building libraries. And there have been instances where you can build libraries of A-TEEM data with fluorescence. That library should capture the variations that you may have in the complex mixtures that you specifically are looking at for your application. And the reason I say this is because fluorescence, tryptophan fluorescence will change with the local structure, the local solvent environment. The peak will change, the intensity will change with concentration, the width will change as well. And so you can have a library entry for tryptophan, but based on the local environment it is, especially in these complex type protein mixtures, you would have to have an entry for everything that you do know. So if you do have pure components, yes, I don't see why a library couldn't be used. And this is kind of part of the A-TEEM method development, right? If you have an A-TEEM method and you have pure components, this is the best way to build your calibration data set and multivariate analysis. And then to use your samples to try to validate that as well. So I guess the purpose of a library, you have to really understand the variation in your fluorescent materials and molecules to be able to build a library appropriately. But again, this is all part of A-TEEM method development that we're doing with every single one of the applications that I presented. Now, the other one was to characterize the exposure if components are solubility limited. Yes. So again, anything, especially for proteins and cell media, I think you're talking about like water exposure. So if you have something that's kind of hydrophobic, yeah, the fluorescence is going to be different, whether it's exposed to water or whether it's hidden in a hydrophobic pocket of a protein. And there's many studies based on exactly using tryptophan and tyrosine for exactly that, especially tryptophan. The fluorescence changes definitely based on exposure. So again, you would have to have a range of samples where that exposure is controlled in order to do a test on that or to build a library for that for your calibration set. I hope that answers the question.
Very good. Thank you. Now, you were touching on this while this attendee was writing this, but they're asking if you could comment on if someone would want to use A-TEEM to characterize an in vitro bioassay exposure. Does A-TEEM have sensitivity to differentiate C-free versus C-bound, where bound could be bound to proteins in the serum or bound within cells of the assay?
C-free versus C-bound. Yes, it maybe. I would say maybe. So again, this is part of the A-TEEM method development. So we would have to try it. But the proteins, you know, again, the fluorescence from proteins changes based on local environment. So if you have a protein that's changing in local environment based on whether it's in the serum or bound, I would say there's definitely going to be a difference. But we would, again, have to try it based on a set of standards, based on a set of expected samples or samples that kind of explain variance of that solution.
We're almost out of time, but I want to squeeze in a few more questions. There's some great ones coming in. So our next question, let's see. What do you know about using the Aqualog for chlorophyll A fluorescence?
Chlorophyll A fluorescence. Yeah. Okay. So we've had some customers using I don't know if it was the Aqualog, it maybe have been one of our other instruments, but using fluorescence signals to characterize chlorophyll A. I don't believe it was done using EAMs or A-TEAMs, though. These were single spectra looking at ratiometric fluorescence for chlorophyll A. I would definitely encourage you to contact our fluorescence applications group in New Jersey, because we do have some, I don't know if we've finished our application note on this, but I do know we have experience for that. Chlorophyll A has, you know, 2 different fluorescent peaks. And so based on how those change, you could probably tell the, you know, the state of your chlorophyll molecules based on the A-TEAM. But it may not be needed, as it's depending on it really depends on the complexity. If you have other molecules that are fluorescing and overlapping with the chlorophyll, then A-TEAM could be a way to go. If you don't, then 2D fluorescence may be a way to go as well. So it really depends on the application there.
Very good, thank you. Now, sometimes we see an overlap of multiple components in the contour plots. Is it the Parafac or any other analysis that can help differentiate the signals?
Yeah, that's a good question. So yes, Parafac is specifically to distinguish overlapping signals with which are broad. Fluorescence is a broad spectrum. It's not like Raman or FTIR, which can have very structured and very narrow peaks in comparison. And so parallel factor analysis can absolutely help differentiate the signals that are overlapping. Tryptophan and tyrosine overlap quite a bit, actually, depending on their environment. And so we use this for Parafac quite easily. Yep.
Now we have more than 2 left, but I'm going to ask these last 2. How would one know what should be the shortest excitation wavelength for their sample?
Great. Yeah. So the shortest excitation wavelength for your sample is where your sample is absorbing and if that region is of interest to you. So to measure an A-TEAM plot, the Aqualog can excite down to 200 nanometers. It has a ozone-producing xenon lamp, and up to 800 nanometers. And so in that region, if your sample is not absorbing and there's no data to be gained, that's the region that you don't need to use. And you can select to excite above that or below a region that doesn't have any absorbance in it. Yep.
Thank you. And we'll wrap with this final question. Does the number of samples being analyzed affect the reliability of the multivariate analysis? Is there a minimum number?
Okay. Yeah, that's a really good question too. Yes. I mean, the more data, the more statistics you have, the more reliable your model results will be. But just as importantly is you need a number of samples, a number of data sets that is going to accurately represent the range of data that you want to analyze, right? So if you have concentrations that range from 1 to 1,000 parts per million, or whatever the units may be, you're going to need enough samples within that range to describe your data accurately. And that's where the minimum number is not, it's not a more or less. More is going to give you more statistics, but you want to accurately sample the range of the variance that you want to describe. And that's what becomes more important.
Dr. Gall, thank you so much for your time and your valuable information. Do you have any final comments for our audience?
No, I want to thank you for your attendance, and thank you to LabRoots for hosting us today. And if you have any questions, you can contact HORIBA or go to our website directly. So thank you.
Thank you again. And thank you to our sponsor, HORIBA, for helping bring this educational webcast to everybody. Now, before we go, I do want to thank the audience for joining us today and for their interesting questions. There were a number of questions that we were unable to get to today. So as a reminder, those questions that we didn't answer today, and additionally, those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you for joining us, everyone. Until next time, have a great day.
Thank you.